Hi, good day to everyone. My name is Nurul Aiza binti Muhammad Sabri. I'm from Utah Sungai Long campus. So my project title is Modulation of Endothelial Nitric Oxide Synthase by Epigallocatechin Gallate (EGCG) for the treatment of vascular dysfunction in angiotensin II-infused hypertensive mice. So before we go any further, this is my background of the study. So hypertension is a condition in which the blood vessel have a persistently raised of blood pressure. So according to the cardiovascular disease, 17.9 million people died from CVDs in cardiovascular disease in 2019, which representing 32% of total global deaths. So in Malaysia, until 2020, CVDs remain as a principal cause of death. So this is a blood pressure stages by American Heart Association. So the pathophysiological of hypertension is complex and is the most common cardiovascular disease, cardiovascular risk factor, which can contribute to the widespread morbidity and mortality. So maintaining the blood pressure within the normal range is depend on the balance between cardiac output and also peripheral vascular resistance. So endothelial cells line inside every blood vessel. They form a one cell thick layer called endothelium. Endothelium is a multifunctional organ actively involved in the modulating vascular tone and structure and also regulates vascular homeostasis. So a healthy endothelium is able to respond in physical and chemical signals by producing endothelium-derived relaxing factor, which EDRF, and also endothelium-derived contracting factor, which is EDCF. So the decrease in the release of EDRF and also enhanced production of EDCF results in the dysfunctional endothelium, which will leading to the various cardiovascular disease. So since endothelial dysfunction and CVDs are closely associated, so treatment to address endothelium dysfunction has been a major focus in the prevention of vascular complication. So in recent years, there is a growing interest in the usage of um, medicinal plants such as green tea, such as tea, to as an alternative management of CVDs. So green tea is a type of is a type of tea made from Camellia sinensis leaf. It is established that green tea uh, contains a wide variety of compound such as catechins. So in this study, we are focusing on the epigallocatechin gallate (EGCG) to see their benefits on the cardiovascular disease. So there's a numerous studies that study about the action of active product in tea in protecting the endothelial function of hypertension. So that's why in this we are design our this study um, to 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 see the behind the mechanism of EGCG to protect the endothelial dysfunction. So the objective in this study is to investigate if EGCG treatment ameliorates a eNOS uncoupling and thus improve the endothelial function of angiotensin II-infused hypertensive mice. So briefly, this is a flowchart of my research project. So the animals will be the animals are divided into four groups, which is control treated, angiotensin 2, and angiotensin 2 treated. So azot osmotic mini pump will loaded with angiotensin 2 will be implanted subcutaneously into the animals, and then uh, the EGCG treatment was given to the animal for 2 weeks. And then during the 14 days of treatment, the systolic blood pressure was measured by using tail cuff method. So at the end of the treatment period, all mice will be sacrificed by carbon dioxide inhalation, and blood sample from the animals were collected, and the thoracic aorta were isolated and placed in a Krebs solution. So some of the vascular tissue was snap frozen using liquid nitrogen and stored in the -80 for later processing. So we move to the results part. So the first result is systolic blood pressure. So the basal systolic blood pressure for all animals was similar. And infusion with angiotensin 2 increased the systolic blood pressure of the animals starting on the second day. And this increase was continuous in angiotensin 2 infused hypertensive mice without EGCG treatment. But for the angiotensin 2 group treated with EGCG, there is also increase in systolic blood pressure, but the increase is attenuated starting on the sixth day of treatment by EGCG. So this is the vascular activity study. So there was a decrease. From this result, it's demonstrated that there was a decrease in relaxation to acetylcholine in aortic rings of angiotensin 2 infused hypertensive mice compared to the control group, which exhibited more than 80% of relaxation. So angiotensin 2 infused hypertensive mice which receive treatment with EGCG, there was an approximately 20% improvement in the impaired acetylcholine-induced relaxation. So this vascular functional study also analyzed by using area under the curve. So from the area under the curve results, we can see there's a significant difference between the angiotensin 2 infused hypertensive mice with and without EGCG treatment. So we move to the plasma nitrate measurement. So this plasma nitrate level measurement are by using Grace reagent. So the result shows that the angiotensin 2 exhibited a significantly higher level of plasma nitrate compared to the control group, and 2 weeks treatment with EGCG significantly decreased the level of plasma nitrate in both control and angiotensin 2 infused hypertensive mice. So same goes to the intracellular nitric oxide level, which the angiotensin 2 group exhibited a higher level of NO level compared to the control group, and EGCG treatment decrease the level of NO in decrease the level of NO in vascular tissue. So next, we move to the measurement of total CGMP level by using commercial. This measurement of aortic CGMP level is using Aiza kit. So these results demonstrate that angiotensin 2 group exhibited a significantly lower level of CGMP compared to the control group, and also treatment with EGCG for 2 weeks significantly increased the level of CGMP level. So the result has been demonstrated that CGMP has been in part to help regulating to lower the blood pressure, and also by regulating the smooth muscle cells. Next is a total BH4 level. So this study demonstrate that angiotensin 2 group exhibited a significantly lower level of BH4 compared to the control group, and also treatment with EGCG for 2 weeks able to increase the level of BH4 level. So these results, BH4 results represent a potentially therapeutic agent in regulation of eNOS function in vascular disease. So in RS level, so this result demonstrate that aortic tissue of angiotensin 2 infused hypertensive mice has significantly higher level of ROS compared to the control group, and also treatment with EGCG able to lower down the increase in ROS level, especially in angiotensin 2 infused hypertensive mice. So based on this result, it has been shown that antioxidant properties of EGCG able to inhibit the increase in ROS level. So same goes to this NOX2 result. So this NOX2 is one of the absorbed form of the NADPH oxidase in the aorta of treated animals. So NADPH oxidase has been implicated as an important pro-oxidant enzyme, which can contributing to hypertension. So this finding suggests that the anti-hypertensive effect of EGCG may be attributed at least in part to its antioxidant effect of EGCG by down-regulating vascular NADPH oxidase activity in hypertensive animals. So as a summary for all the results, we may see that the increase in BH4 level helps in the activation of eNOS to produce more NO. So NO will activate soluble guanylate cyclase to CGMP, which then mediate vasodilation. This evidence is in line with the functional study result, whereby the EGCG able to improve the impaired vascularization in angiotensin 2 infused hypertensive mice. But in the presence of NADPH oxidase, The oxidative stress superoxide anion will be generated by NADPH oxidase, and ROS will reacted with NO to form peroxynitrate. So this will further oxidize the BH4 and decrease the NO bioavailability. A decrease in NO bioavailability in the vascular system increase the oxidative stress and therefore promoting the endothelial dysfunction. So in the presence of EGCG, it helps to increase the level of BH4 and CGMP level, which further helps in restore bioavailability of NO and thus improve the vascular dysfunction. So conclusion from this study, overall collectively observation from this study suggests that EGCG may potentially be useful for the treatment of hypertension by improving the endothelial dysfunction. That's all from me. Thank you.